This final lecture related to data is on reproducibility. It is in Module 1, Data Science. In this course, we will use Microsoft Excel. The spreadsheet layout of Excel is similar to other statistical softwares. Data in Excel are arranged in rows and columns. Each row represents a subject. Each column represents a variable. Each cell contains the particular value that the subject takes for that variable. A cell is an intersection of a column and a row. Cells are called by the column letter, then the row number. In this example, cell B1 is selected. You can see that B1 also appears directly below the ribbon. It is important for data to be arranged clearly and concisely so that others who may use our information in the future are able to reach the same conclusions. There are a few more terms that we will use often throughout the semester. A parameter is a numerical summary that describes the population. Numerical summaries may include means, medians, standard deviations, ranges, proportions, percentages, odds, and risks. Parameters exist, but we do not know the numerical values that they take. Instead, we denote them with symbols, which are often Greek letters, such as mu or sigma. Remember that parameters describe the population. Both words start with the letter P. A statistic is a numerical summary that describes the sample. Statistics exist, and we can calculate their numerical values by using the data that we collect from the sample. Remember that statistics describe the sample. Both words start with the letter S. Because samples estimate populations, statistics estimate parameters. Basically, this graphic is a summary of statistical inference, which we will talk more about in the next 2 lectures. We want to know about the population, so we use random selection to select our sample. From our sample, we obtain a statistic, in this example, our sample mean, which we can use to estimate the parameter, the population mean. Objectivity is also important in statistics. Objectivity is defined as the intent to measure things as they are without shaping them to conform to a preconceived worldview. Objectivity is important in all aspects of a study, including designing questionnaires, sample selection, and data analysis. When designing questionnaires, one must make sure that the questions do not lead those being surveyed to a specific answer. One should strive for a random sample, or at least a sample that is as unbiased as possible. Data analysis should tell an objective story, not one that the researcher wants to show, but one that is true based on the information. In summary, to obtain an accurate representation of the population, we must do our best to ensure that data collection are useful. However, nothing is perfect, and errors can appear in our data. Imprecision is the inability to get the same result upon replication. Imprecision is often quantifiable, which means that if we take enough measurements, we can estimate how imprecise that particular measurement is and account for it. On the other hand, bias is the tendency to overestimate or underestimate the true value. This can be difficult to quantify, and most of the time we don't know that it's happening. We call a measurement valid if it's unbiased. In the graphic, we see 4 different measurements, one that is precise and unbiased, one that is precise and biased, one that is imprecise and unbiased, and one that is imprecise and biased. We would like our measurement to be precise and unbiased, like the first option. All of the measurements are clustered closely around what the true serum glucose level is. However, even if our measurement is imprecise and unbiased, the third option, we can still get an estimate of what the true serum glucose level is. However, if our measurement is biased, the second or fourth option, we have no sense of what the true serum glucose level is. Therefore, it is worse to have bias as a measurement error than to have imprecision as a measurement error, though we would prefer to have neither.